Hello, my name is Alaa Aziz, and today I'll be talking about kinetic studies on the catalytic mechanism of F420 dependent glucose 6-phosphate dehydrogenase, or in short, FGD. I will start by giving you some background information on FGD. FGD crystal structure was resolved by our collaborator, Dr. Gadar Bashiri. It is a 78 kilodalton homodimer. So if you split it down the middle, you get two identical subunits with an active site with the F420 cofactor bound deeply in the active site of each subunit. This is an expanded view of the active site. We have these three important amino acids: glutamate 109, histidine 40, tryptophan 44, that are believed to play an important role in catalysis. Using docking studies, they were able to model in a glucose 6-phosphate, which revealed important interactions between these three amino acids, the glucose 6-phosphate and the F420 cofactor. The F420 cofactor is spectrophotometrically distinct. The oxidized form shows high absorbance at 420 nanometer. Upon reduction, it loses this high absorbance and gains a shoulder at 320 nanometer. This is what allows us to do the type of chemistry we do in our lab, where we look at the rate of reduction of the F420 cofactor. FGD catalyzes the conversion of glucose 6-phosphate to 6-phosphogluconolactone, and it produces the reduced cofactor. The reduced cofactor plays an important role in the activation of natural emitters of pyrine prodrug that treat multiple drug-resistant and extreme drug-resistant cases of tuberculosis disease. The reduced cofactor sends 2 electrons to an accessory protein, which activates the prodrug. Upon activation, this prodrug is broken down, and a nitric oxide is produced, and which is toxic to the cell. Dr. Mercy Uyugi in our lab was able to update the mechanism. She studied the order of substrate addition. She determined F420 binds first, followed by glucose 6-phosphate. She also determined that glutamate 109 acts as the active site acid using pH profile. However, she wasn't able to determine the active site base that abstract the proton from glucose 6-phosphate and facilitate the hydride transfer. We looked at the additional amino acid in our active site. We found it's histidine 260 and glutamate 13. We mutated those, and we also continued our studies on the histidine 40. Our binding studies suggest that these amino acids play a role in the F420 binding, but not the glucose 6-phosphate binding. Our steady state kinetics showed that these amino acids are pretty much dead. These variants are pretty much dead, suggesting that they play an important role in catalysis. But this doesn't tell us whether or not they are the active site base. We continued our study to pre-steady state kinetics, where we look at the individual steps before the turnover of the reaction to products. We use the self-flow spectrophotometer to do the experiment, where it collect data on a millisecond time scale. And we use Kentucky Explorer to fit the data on here, where we are able to create a model. We know that cofactor binds first, followed by substrate. We took into consideration an intermediate that might turn over in the reaction, and product release step. This experiment told us two things, and one is that we do have an intermediate in the reaction. This is shown by the green line, that it is not completely overlapping with the blue line. And also we have a fast chemistry step, slow product release step. Our pH profile suggests that this histidine 260 is not acting as the active site base, and the glutamate 13 is not acting as the active site base. However, it's assisting somehow in the reaction. We turn our attention to the NADP-dependent glucose 6-phosphate dehydrogenase mechanism, where they have the histidine 240 and aspartate 177 forming a catalytic dyad, where the histidine 240 is acting as an active site acid, and then donating a proton to the aspartate 177, and then acting as an active site base, abstracting a proton from glucose 6-phosphate and facilitating the hydride transfer to the NADP. This got us to think of the role of our glutamate 13 in our reaction. We think that histidine 40 and glutamate 13 are acting as forming a catalytic dyad as well. We did this temperature dependence, where we are able to do a complete pH profile. So each dot here is a complete pH profile at different temperatures. And we fit this data to a Van 't Hoff equation, where we got the delta H of ionization and delta S of ionization. This table right here is a Cleland table that that tells you the expected delta H of ionization for each functional group. Our delta H of ionization fell between 6 and 7.5, so we got 6.3 and 7, which suggests that it's a histidine that is the active site base. However, it's acidic, so we think this histidine is acidic first, acting as an acid first, and then as a base. I would like to thank Dr. Johnson Winners and my lab members.